So I'm going to talk about boosters, Eterna script boosters. And I'll take you through going into the lab and going into a design puzzle page and how you access your boosters and what different things that they can do for you when you're in the game designing things. So if I could, I'll share my screen now. I'm going to actually open play on the home screen of Eterna and come down to the second line where it says labs. Click on that, and I'm going to use the T-Box Switch Pilot lab. It's all closed now, but it's an example so I can show you the boosters and what they do. And I chose this because it's a small molecule and you'll have a lot of room to see what's going on. So I've clicked on the labs. There may be people here that have never gotten into the labs or don't have permission to yet. So maybe this would be good for them to know what they're up against when they finally get there. With every lab, there's a box like this over here on the left, where you see a review or a design. Review brings you a list of everybody who submitted designs to the lab. When you go to design, you'll just get a blank starting sequence and you start designing all on your own. I'm going to go to the review button and bring up a list of what the solutions were that were submitted to the lab. There are a lot of them, and there's almost, I think, 200. I'm going to select one of mine, which is right about here. And it says whbob 8M. So this is the database of everybody who submitted a design, and you can see what the designs are. If you go to the left side and click, a sliding window will come in from the right-hand side. I'm moving it over so that I can see past the individual pictures of people. So here's the view copy to look at that particular lab. Now I'm going to slide the window out of the way too, so I can demonstrate this. So here's the Eterna toolbar at the bottom. And over on the left side is a lightning bolt. And that lightning bolt has several things that you can access. The bottom two are symbols like a rocket ship, pointed at the top and a V at the bottom. The bottom one says copy, delete, replace. The next one up says mutation marker. Those icons, those symbols, represent boosters. And boosters are code written by players just about exclusively, I think, in Eterna script, which is a combination, I think, of HTML and JavaScript put together to create these amazing things. So when you want to call up a code like copy, delete, paste, this booster is, you just click on it and it occurs. Now what this does is it opens up, in effect, an HTML form with input text boxes and buttons, copy, delete, and paste, which when you click one, calls up a JavaScript function and does code. Pretty good magic, I think. So here we have a start button. I'm going to start input. Let me slide this over a little bit. You have the start position and end position of the sequence. And if you put in, say, 1 to 10, and you hit copy, that would start a JavaScript function that would go and find those base locations and give you what's on there. I'll do this just to show you. So there's 1 to 10, copy. There you are. There's your first 10 sequences. I'm not going to go into all the things you could do with this, but basically you've got a way to copy, delete, or replace any sequence that you're working in the lab. In effect, you have an RNA editing tool right here. It's amazing. And it takes your input, takes your decision-making buttons, and gives you back an output. So that's one type of a booster that is interactive with the player. To get rid of it, I've only found a way to reload up here on the left-hand side, to click the reload and then go back into the design again. So I've done that. I hit the reload. And I'll come back to the same design. Click the view and move this to get this over out of the way. So you can see the booster is gone now, and you're in another design. So I'm going to show you the second type of booster. It's called the mutation marker. Same thing, you go to the lightning bolt tool option, find your booster, left click on it. In this case, there's no external input you have to do. The actual code is asked the question, how many mutations have you made and what are they? So if you were to look here at this terminator loop in STEM, you can see this GU pair, this GU pair, and a U here. They have black circles around them. Those are the mutations. Not only does it tell you which base has been mutated by using the black circle around it, it also tells you what it used to be and what it is now. Here we are at 206, location 206, and it's an A, adenine, and it used to be a guanine. See that little red section on the side of it? I don't know if that's too small for you to see. So it not only tells you what's been mutated, it tells you what it used to be and what you've changed it to. Now this happens to be a switch that has two states, and this is state number 1. This is state number 2. You might see the mutations a little bit easier here with this. There are 2 mutations here where they were changed from GCs to AUs, weakening this string. So I don't want to get too deep into this as far as the game is concerned, but it's really amazing what you can do with these boosters and the information you can get out of them while you're playing. Wow, something's happened. My mouse is freezing. Yeah, I don't know what's happened. There we go. Okay, so I shall get out of this again. Yeah. Okay, so I've shown you how you can find your boosters if you already have them, but obviously if you don't have them yet, I need to show you what you can do with this. Again, for some reason, I'm having some problems with my mouse. Okay, I've switched over to what's called the script homepage. And this is where all the scripts exist that you could choose to use in your designs. Notice up the top here, we have something called favorites with a star. If you press that and you have any favorites, they'll show up in the example. So you see, you can hear a star booster for each of these. The two boosters I just showed you on the design screen, these are these boosters in the scripts page. So let me go back. And this is called the home page here. I just clicked favorites and it can show me just my favorites. There they are, just my favorites. Now, let me come down here. And I'm looking for one of Astro's designs. Right here. It says Booster Astro's First Solvers. This is a design, Carosi, I think it is. Yeah, Carosi made this into a booster, but it's not one of my boosters. All I have to do to make it one of my boosters is to access the actual Eterna script. And this is what the Eterna script page looks like. This is the code right here. And if I want to use that, then I come to the title page, and I go over to favorites with a star and click on it. It's changed to unfavorite. So when it was favorite and I clicked it, it became a favorite. If I didn't want it to be a favorite anymore, you would click the unfavorite and it would become not there anymore. So now that I've done this and made this my favorite, I'm going to go back again to the home screen, labs, do the review, come down to that same design, right here. Now, I'm going to check this off so you can view it. Just like before, we're looking at an individual design, and the boosters before were working on their own design. Whatever design you were in, it was just working on that one sequence. But with this, let's see if I can find my, here it is right here, Astro's First Solvers. So I'm going to click on that and you'll see what happens. Boom. Right up here over on the left is the first solvers. Digitalembrace was the very first solver, the very first submission to this lab. Now this isn't doing something about this one molecule, this one design. This is getting information from the entire lab set of solutions and giving you the data on every one of them. In this case, we're only looking for who the player name was of each design. And it basically is taking from the lowest design number to the highest design number and listing them out here. So in this case, you're doing analysis on the entire lab and not just this one sequence. It's pretty amazing. You can come up with that kind of information in a split second. And you can do it on energy levels and all sorts of other things. So that's pretty much it about what you need to do for a booster. You want to make it a favorite using the script section. You have to go to the home page and you have to select one of the things. Now see here, here's the line for Booster Astro's First Solver. It's now got a star on it because when I made it a favorite, that's the symbol that denotes that it's a favorite of yours. And it's only your data that's going to show there. It's not going to work for anybody else until they make that particular script a favorite of theirs. And then they can have it come up on their design page. And that's it, Jonathan, I think.
All right. That was great. Thanks so much, Bob. So next up is me. I'm going to talk about 2 spreadsheets that were made by players during the Ribosome challenge. So I'll share my screen here. So I'm going to talk about the ribosome results spreadsheet, which most of you have already seen, but I want to talk about that really briefly. And then I'm going to talk about the motif protein contact boosters that Omi, Eli, and I made during the ribosome challenge. So here's the different links, and Slava's going to post those in the chat as I'm talking about them in case anybody wants to look at those docs. So was there a question? No. Okay.
No, sorry.
Okay. For the ribosome challenge, we had gotten the results from the pilot round and the results from round 1, and I was looking at the results and looking at the different designs and trying to figure out. I was trying to wrap my head around which designs did well and which didn't. I was wanting to compare different characteristics, and I thought, well, I'm going to put them all in a spreadsheet so I can look at them. So this is what I made, and I made there for each round. There's a 16S and a 23S, and I made 2 sheets for each sub puzzle. So for example, here, this is the first page I made for each puzzle. I put the ID for the design, and it has a link here into the design in the game as well. And then here is a list of all the mutations in each design. And then I eventually made a final sheet that had all the mutations compiled. So it was easy to search it and see if a mutation had already been made. And then on the second sheet, again, I did the design ID with the link, how many mutations, and then I wrote a short characterization of what each design was doing, what approach it was using, so that we could quickly look through what everything had done. And then I noted if it had any motif violations, any protein contact violations, any IUPAC violation. The IUPAC is conservation data from gamma proteobacteria. And then also I noted whether it deleted any repeats, because we've been testing this idea that maybe the repeats have been causing an issue during the experimental process. So players started using this, and we all really liked it. It was quite interesting to use, but it was rather tedious. Well, no, never mind. That's not what I wanted to say. Okay. Yeah, so everyone's used this. That's enough on that. So the second thing we made was when we started the ribosome challenge, Eli started talking about motifs in the ribosome, structural motifs, and also protein contacts, and that we should be probably aware of those, that mutating those and changing those might be causing the ribosome not to form, not to fold, or not to function very well. So Eli created this sheet. He got the locations from Riju, and Riju and Andy wrote a piece of software that's available on Rosie, and they ran it with whbob and got a list of the motifs and proteins and gave those to Eli. And he created this spreadsheet. And it basically looks like this. So for each, we have the base positions on the side here. So if you were thinking about mutating base 71, you could come on here and see, oh, there's an A minor motif there potentially a platform. And then further over on the right, we have all the protein contacts as well. And this is where I wanted to say this became rather tedious to be working in the game and then have to go look up each position one by one. It was very time consuming. And so we started talking about, could we make a booster for in the game? And also, I believe after round 2, I talked to Andy and wondered if we could open up any of those protein contacts. Somehow there was a discussion, and he was talking about the backbone contacts, and then that if it's only backbone contact, it's probably still okay to mutate it or to do a purine mutation or pyrimine mutation. So he went through the crystallography and looked all those up for us and gave us the list of what he judged to be some of the safer mutations we could potentially make. So we took all that information, and Omi started a big spreadsheet, and I had a sheet for the motifs, a sheet for the protein contacts, and then a third sheet for other contacts. And we all took the data that we had of what we knew and brought it over to this big spreadsheet. And I remember spending several days doing, we had to go in and do each entry line by line, putting it in there. And so it took a lot. It was a huge job doing all this and entering it in there. And then Omi wrote up the code and showed us how to do each kind of coding for whether we wanted, whether the mutation was, didn't want to allow it at all, or whether there could potentially be mutations to those sites. And then let me put that all on one sheet, and he wrote a booster script, exported the data from his spreadsheet, and then imported that into the booster script. And he's got it working, and it's been working really great starting in round 3. So I'm going to show that now. This is an example from that large spreadsheet. So for the A minor motifs and UA handle here, for example, we didn't put any allowed mutations. So if somebody clicked on something and it was a UA handle, which just said, here's a flag, there's a UA handle there, you probably don't want to touch that. Whereas on these proteins, we indicated that C and U are okay. So if it was a C and you changed it to a U, the player would not get a flag saying, be careful here. And also you see a G and RA, which is a well-known structural motif. So for the G, we say, hey, this needs to say a G. The A, this needs to say an A. The second one is the N, that can be anything. And then the R can be an A or a G. And here's what it looks like in the game. This is one of my designs from round 4. So I mutated 906. It came up in the booster list, which is when you hover over this, you see the motif proteins list here. And I'll say, Omi made this an in-game booster, so you don't have to add it to your list and call it up. It just automatically is there. And I'll also note that I added an annotation on this and noted, okay, I know that's a platform B motif, and I got this mutation from Thermus thermophilus. So some key points I'll highlight is I want to show researchers the kind of great, really unique, interesting things that players do both in the game and also to analyze data afterwards. And I want to show players that how we can all come together and analyze data. And there's some simple tools, especially these Google Sheets, that anyone can use. If you see something and you say, I'd really like to see this data presented in a certain way, I mean, go for it. I am not a spreadsheet expert, or I wasn't. I watched YouTube videos that showed me how to do things, and if I needed to do a specific thing, I would Google that. And then Omi helped me because Omi's very good with spreadsheets. And then up next is Amy, who we call our spreadsheet queen, and she's quite good with formulas. And also highlighted in my first spreadsheet how having the design notes in when people are making submissions can be very helpful for gathering that information for us to look over for data analysis after we get results back. Also want to tell people it's fine finding these external tools also. It can be creative and go out and find other tools. Like right now on the pseudonaut, we're using databases of known pseudonauts to use for testing. I want to note the protein contacts that Andy did. He has verified all those in 7K00, which the Kate Lab, I believe, is who made that crystallography. But the motifs have not been verified. So if any graduate student or someone wants to take that on as a project, we'd love to have those verified. You probably also noticed in the game the IUPAC seed booster that covered the gamma proteobacteria conservation that we've been using to help us guide for designs. And I want to acknowledge that J. Anderson Lee largely created that along with Master Stormer's help. If you have any questions, you can email me at this address. And I think that's it. I will stop sharing. There we go. Are there any questions, Jonathan?
Nope.
Yeah, it was a long process. Joseph, it was like a year, at least a year, compiling all that and getting the work done. Yeah. Okay, so up next we have Amy, who's also going to talk about a couple spreadsheets that she's been using for organizing and analyzing data.
All right, I'll share my screen. So I call my little talk the joy of spreadsheets, how you can find spreadsheets to be useful in Eterna. Okay, here I have a spreadsheet with some data from the Ribosome Challenge round 3. And these are all the designs. And this actually goes down for about 200 rows because that's how many designs there were in that lab. And you can see there's a lot of data here. We have the player and the title and the ID. We have the expected accuracy, the violations for IUPAC or protein motive, the number of changes, the minimum free energy, delta, and design approach. Now, when you have a huge spreadsheet with 200 lines and all this data, it's really hard to figure out anything useful from it. So there's ways to use spreadsheets to organize the data and find some good information. So first, I highlighted the column. So in Excel or in Google Sheets, there's a simple command that's called conditional formatting. It's called the same thing in both programs. And you just click on that and you can choose to make the highest number green and the lowest number red, or other ways of highlighting. So here I highlighted the EAs with the highest in green, the lowest in red, because we thought highest EAs were the best. And then the violations, I just made it red whenever there was a violation. For these last three, I highlighted them with the lowest numbers in green and the highest numbers in red. So you can see we can now look at a row and kind of see how much is green, how much is red, and that might give us some more information about how good we think a design is. But there's still more we can do. We can sort the columns. So we use the sort and filter command. And in Google Sheets, it's called filter data and filter views. And it puts a little arrow in each column. When you click on that arrow, it sorts the EA, it sorts that column, and you can tell it highest to lowest, lowest to highest, or alphabetically, however you want to sort it. So here I've sorted by EA. So the highest EA values are at the top going down. And so we can see right away the number of changes is now, all of these are pretty red, pretty orange. It's easy now to go down and see which ones had the least number of changes for these high EAs. But gee, in this lab, I wanted to only have like 20 or 30 changes, not 154. That's just too many. So there's the next thing I can do is filter. So I use these same little triangles that we put in with the sort and filter command. And I click on it and it allows me to filter by whatever number. So I'll filter the number of changes between 20 and 40. So this gives me all the changes between 20 and 40. And the EAs are still sorted with the highest ones at top and the lowest ones at the bottom. So I can right away see for a certain number of changes, what was the highest EA and did it have any violations? So this is just a way of organizing a huge amount of data to find useful information out of it. Next, I have a different spreadsheet. This spreadsheet came from Open Vaccine Round 6, and it has data in it. And I'm going to look for correlations between the data and the design parameters. Now, there's actual data underneath these columns. I just kept it hidden for intellectual property purposes, but the data has already been highlighted with the highest in green and the lowest in red. And I sorted by half-life. So the highest half-lives are at the top. The other columns in the spreadsheet show that some of the parameters, some of the measurements that were made of each design, the DEG score, the free energy, the SUP, which was some unpaired probability. We have some measurements that had to do with the branchiness, the mean ladder distance, the number of hairpins, and the number of junctions with 5 branches or more. And then also I added the GC content. So we can see when we sort by half-life with the best at the top. Oh, and the parameters I all highlighted with light blue as the lowest numbers and this purplish red as the highest numbers, just to differentiate the parameters from the results. So we can see DEG score gets higher as a half-life gets lower. So that shows, well, those might correlate. So the half-life might be dependent on the DEG score. And we can look at that mathematically in Excel or in Google Sheets using a correlation function. So in Excel, on the data tab, there's this data analysis routine, and you can choose your whole table, and it'll show you the correlation of each column with each other column. Now I reduced it to only show correlations of the 3 results that we're looking at with the different parameters. And we can see, oh, and a correlation number, a 1.0 is a perfect correlation. That means all the result is dependent on whatever the parameter was. And a negative 1 is a negative perfect correlation, which means your result would increase as your parameter decreases, but it's still a really good correlation for a negative 1. And a 0 is the worst correlation. So something like this is not correlated at all. So here we have, for the half-life, these 3 parameters had the best correlations, and we can kind of see that with the way the colors get more purple. And then I looked at the same thing for the 6-hour expression. That was the protein expression telling us how much protein was made after 6 hours. And so as the expression got better, the mean ladder distance got lower, and the number of hairpins got higher, and the number of junctions with 5 branches or more, that's big multi-loops, got higher. So we could see good correlations here with all the branchiness parameters when we look at 6-hour expression. And mathematically over here, they also showed good correlations. So this just shows some of the things that you can do with spreadsheets without too much trouble, just to try and find out how the results are correlated to the parameters. Now I didn't add a slide for 24-hour expression, but you can see it was very similar to the half-life, probably because only the RNAs that lived long enough were expressing at 24 hours. So the ones that were expressing more at 6 hours to give these better correlations were probably not there anymore at 24 hours. Okay. Oh, and then we can make graphs. So you can make graphs in Excel, and it's really pretty simple. All you have to do is you go in your home tab, you go to analyze data, and you can type in half-life by free energy, and out will pop a graph. Or any of these others. These were the ones that had the best correlations. And the graphs can sometimes help you see how the correlation, like here we can see, well, there were a lot of them with a small half-life, but the ones with the best half-lives all had lower free energies. Okay, now something slightly different that you can do with a spreadsheet is extract information from the sequence of bases. So this spreadsheet is our designs from Roll Your Own Structure, and the list goes on for like 1,000 entries. There were lots of designs in Roll Your Own Structure, and we have the sequence. It was a fairly short RNA, so that's the whole sequence. And over here is the dot bracket structure. And if you're not familiar with that, the dots are unpaired bases, and the parentheses or brackets are the paired bases, and it kind of shows which ones are paired with which. And so that really defines the structure, defines the whole structure that we see on the screen in Eterna. So what I wanted to do in Roll Your Own Structure was find all the tri-loops and figure out which tri-loops performed better. So I had this huge spreadsheet of data, and I wanted to find all the tri-loops. How could I do that? Well, there's a command in Excel for search. And I know a try loop was a ... And I put 2 brackets around it, so I'd have 2 closing pairs on my try loop. So it's a nice stable try loop. And then I just searched this structure column for that string. That's called a string. And it returned a number here that tells me where that string existed within the structure. So it was at position 11. So right here at position 11, you see the try loop. And then I wanted to get the try loop sequence. So I go over to my sequence column and I use this command to find the 11th position over. And I had a GAA at the 11th position. And so that came out as what my try loop sequence was. And then I could also pull out the closing pairs in a similar way. So I know if it's a GC or a CG closing pair. Then I wanted to know the degradation score. Now the spreadsheet had a lot more columns going out here that actually had the deg score and the shape score for every base in the sequence. So this more complicated formula down here would find the maximum base score of the 3 bases in the try loop. And then I use that max score to determine which try loops were most likely to degrade. And I colored those red and the medium ones yellow and the good ones green. So at the end, I could come up with a list of all the try loops that were tested. Some of them weren't tested. And which ones were good and which ones were medium and which ones were poor performers. And it turns out that most try loops aren't very good, but there were some that were good, but none of them were consistently good. But if you want to see more of that analysis, it's at this link, or I'll send some links out after the talk. Okay, so how can you, or how can anyone figure out how to do all this? How did I figure out how to do all this? Well, I have a son who's a computer scientist. I used to ask him lots of questions about my computer. And finally, one day he said to me, "Mother, if you want to know something about your computer, just Google it." So that's what you do. You Google it or use your browser of choice. And you put something in like, how do I find a value within a string in Google Sheets? And you'll find these people have written all these websites that tell you how to do things, or their videos. So it's really not too hard to figure out how to manipulate spreadsheets. So I hope you all find ways to have fun with spreadsheets in Eterna. And you can also use spreadsheets for other things in everyday life, like travel planning or buying a car, keeping records in your garden or your medical records, even grocery lists. So hope everyone can learn to enjoy spreadsheets in Eterna and every day. And thank you.
That was great, Amy. I've been thinking about correlation analysis. I wasn't expecting you to talk about that, so that was very useful. All right, well, I don't see any questions. Oh, Kinwolf has one. Would we be able to export data from lab tables directly to spreadsheet? Well, we don't have lab tables. I don't know if Jonathan, go ahead, Amy.
There are some scripts, some of the data I got from scripts, and scripts can transfer some of the data, but not all of it. Like I think it would do the minimum free energy and maybe the DEG score and the ones that had a DEG score, but a lot of the data, you can't transfer it. And it puts it in a text file, and then you can copy the text file into a spreadsheet.
That's definitely something that we could improve on, is making it easier. I mean, we want to build some tools to be able to do more analysis, right, within Eterna, but also, making the data itself more easily accessible. There's a few different, I think, confounding factors that you wind up running into between just, I mean, it used to be that if you saw it in the data browser, I think at one point in one version of it, there was a little button that you could export it to a CSV or something like that. But even then, there's a lot of data that we don't really store, and it's just kind of computed on the fly. And that's one of the things that we're going to be having to think about how we want to handle it and make it easier to work with that data.
Yeah, a lot of the experimental result data that we're given from the researchers is they generate it and put it in a document that we then, it's in a text, it's textually listed. It's not in the game itself. I think some of what Amy was using there was actually a spreadsheet that the researchers shared with us.
Right,
So-
yeah, the one for with results. Well, actually, also the roll your own structure. I mean, those results came from the researchers.
Dusun shared that, yeah.
Yeah, and I know also getting research data into Eterna and all in one place, that's another area that we're going to be looking to improve on.
Right. I see Slava asked a question about the approach. Yes, the approach was based on the player descriptions, and we had a few approaches that we itemized and tried to fit in an approach for each design that we could.
Okay, I don't see any more questions. Do you, Jonathan? Or people can raise their hand if they want to ask a question. We have a few minutes. Jennifer has her hand up.
So with the scripts, I've had some thoughts. I had to step away briefly during the presentation, but Jonathan had mentioned a while ago that you can call scripts from within scripts. And I've wondered about creating like a set of tools, like this whole funness I've been dealing with the displaying that data in the other window. You know, like if we had a script, once we accomplish that, we had a script that called that for players could use. A lot of common things, so easier for players to do stuff. Just a thought that popped in my head.
Definitely fair point for sure. And there's some stuff that we could do better by actually providing more things by default, but definitely for what we have now, can definitely be really powerful to just, you know, create your own kind of libraries to be able to work with.
And I'll note on scripts, I did create a wiki page on booster scripts, explains how to favorite them. And I put down 5 scripts that I find useful, but if anybody else ever wants to start a wiki page and list scripts, right now it's kind of, you just have to hear from somebody like, hey, try this script, or you can try to go search. I want to point that out too. I think Astromon has his hand up.
Are there any projects being done for the T-Box? Like projects like these tools Amy was talking about in the works?
Not that I know of, and we don't have data from the T-Box yet. I don't know when we will. Are you primarily asking about data, or did you have something else in mind?
I guess infrastructure for that when the data comes out, or
yeah, we're not sure what kind of data we're going to get. Like what probably gonna be protein. I don't even remember right now. I have to look and think about what. I don't want to say it wrong.
thank you.
No, there's nothing in the works that I'm aware of.
There will be once we get a chance to see what kind of data. We'll have some idea of how to make sense out of it.
Hi, Alana. She's head of CGEM, is with us today. And she said she hopes to have experimental data from T-Box in the near future. Thank you, Alana, for the update. Now, Vasco has a question. Go ahead, Vasco.
Hello. I first of all just want to say, hey, I'm very, very new to the community. I've recently started playing, and I think I'm player 8,000. I think on the player boards, and I don't know if that's good or bad. Honestly, I'm very, very new, very green. But I do have. I was speaking to Riju about this probably a week or 2 ago. But I was thinking about publishing or writing a small piece on some of the mechanisms of the methodologies that are used in Eterna as something like 1,500 words. And I probably wouldn't publish it anywhere like NCBI or PubMed or anything like that. But it's something for a capstone project for the certification that I'm doing. And I was wondering what the limitations and kind of what is allowed and what isn't allowed when it comes to publishing or interpreting data, because is the data open source, like is the data open to the public or to the players in the community, And from there, are you allowed to assay it and survey it and kind of code it and interpret it however you want? And is that then allowed to be published under your name or with co-authors? Because I know that Eterna itself has a whole publications board, and I'm not sure if there's steps that I'm circumventing or that I'm illegally like doing if I want to write this, that, or the other thing. And it's just, I don't know. The question that I'm asking right now is I'm blundering a little bit right now, but what is I guess, what are the laws or the rules when it comes to publishing or writing about certain things about Eterna? Like, is it possible?
Yeah, totally valid question. Jonathan, do you want to take that?
Yeah, I can talk about that a little bit. I think the main limiting factor for this is experimental data, right? If the experimental data wasn't there, to some degree, do whatever you want. If you actually look at our EULA, we spell this out, our terms of service. So as is, we consider Eterna designs to be just publicly available. We have some technical limitations on that, but that's really more specific to because of how it's tied to experimental data. But if you want to do any sort of analysis or write-ups or anything, that's awesome. We totally encourage it. The main limiting factor is experimental data, and in particular, when it's unpublished. Typically, when research actually is finished, that data is made available with the papers that get published, right? And at that point, the data is publicly available, and you can, share it and use it however you want. But if it's part of unpublished research, oftentimes that data is only basically made available to the Eterna community before the paper is actually available and the data is publicized. And that's that the experimental data itself is owned by whatever experimental partner we're working with, whether that's the DAS lab or our partners at CGEM or whatnot. So really just you need to make sure that you're not now sharing data that hasn't been released yet.
Absolutely.
Of course, if you want to just write something up and share it within Eterna, please do. We have a section in our forums for labs, right? And we specifically set that up so that you can't necessarily immediately search it from the internet, right? You have to be logged in order to see it. But if you want to just post it in there, no worries for doing that, and we're always happy to see analysis and stuff.
Do you know which puzzle in particular you're going to be using data from?
No, not at all. I've just
Okay.
recently found out what a DEG score is, and I almost have access to the labs. I think I have 1 or 2 more puzzles before I actually do have access, but like that is how green I am. So it's still the whole project and idea is still within its infancy in my mind. But from what I understand, the difference between accessible or non-accessible or publishable and non-publishable is whether or not it was already practiced in wet chemistry or in bench chemistry.
Well, it's whether it's already been published in a paper. If it hasn't, it's still private data that we need to keep in-house. I mean, there's lots of reasons for that.
Well,
One thing
either
is
published in a paper or provided publicly somewhere else, right? Like a lot of Eterna's early data is submitted to the RMDB, right? If it's in the RMDB, it's public. So that's also fine, even if it's not necessarily specifically in a paper.
Yeah. So once you write what your piece, you could ask me or Jonathan, and we'll let you know whether it's accessible.
Absolutely, I will. I am not trying to step on any toes, and the
No,
community.
I'm just saying that would probably be the quickest, easiest. Leonard has something he'd like to add.
Yeah, on behalf of the Players Alliance, which extensively worked on the issue of intellectual property, most of what you're planning to do, Vasco, say your name.
Vasco, yeah.
Vasco, thank you. Okay, most of what you're planning to do, Jill and Jonathan have already explained, which is fine. But I just want to refer to the tools that you saw here. And it seems to me that if someone has shared their own analytics with the Eterna community, they're making that available, but that, to me, making it available in the Eterna community is not the same as saying you can republish it sans attribution. So it seems to me that if you want to say, oh, you know, Amy has got these tremendous correlation matrices that she's put together that you need to attribute what she's done to give her appropriate credit, A, and B, I would also, to be sure of things, ask her permission in this case, if she's comfortable with you referring to that and republishing it somewhere outside of our community. So, you know, I think the general expectation is when players develop these tools and share them in our community, they want them to be used. And I think that's fine. But I would understand here that the players are adding their own intellectual property value when they do these extra analytics, and we should be respectful of that and do attribution and permission. And good luck with it. Thanks.
No, thank you. Yeah, of
For
course.
sure,
yeah.
very good point.
All right, thank you. Yeah. And I just also want to say that the community has been very lovely and everyone's been very nice, and I can't wait to continue the work. So yeah.
We're glad to have you.
I don't see any more questions. So I think we can go ahead and break, and we're gonna have a nice long hour break now.